This conference will now be recorded.
Good afternoon, Saundarya.
Good afternoon.
Very good, Saundarya. Mute yourself immediately. Hi, Tejaswini,
Hello, ma'am.
Suchitra. All are doing good.
Yes, ma'am.
Very good.
Yes, ma'am.
So, any doubt regarding the drug excipient interaction topic, what we covered? I have not completed still. Please mute yourself, avoid that unwanted noises. So if you have any particular doubt, and just quick revision I'm going to do. We started with the one of the topic of part of your first chapter, that is drug excipient interaction. That is a important step we need to carry out in the pre-formulation studies. Hope you understood. It's like what is pre-formulation studies, we got idea. We are not discussing pre-formulation here, but one of the procedure what we follow in the pre-formulation, that's what I was just highlighting last class, that is particularly drug excipient interaction, and that is required very much to minimize the expenses incurred or the overall time which we are wasting unnecessarily to identify the excipients. So drug excipient interaction has that advantage, and of course ultimately it is a stable product which you're going to get out of the all this pre-formulation studies. So in that drug excipient interactions, I even highlighted about what are excipients, what are its role. It adds a functionality to the dosage form, or even for the pre-formulation uh, factors also is affected by the presence of the drug excipient, and also gave idea about the what all the interaction can cause, what is that it comes out after the interaction, what all it can affect, and later on what are the parameters we need to focus on also was discussed. And I started with the methodology also very briefly. I told it's a simple procedure, as I was telling, blending the drug and excipient in a particular ratio of acceptable ratio, and then subjecting into the accelerated stability studies to carry out the interaction or assessing the interaction. So fine. So with that, the drug excipient interaction, Ashita, kindly mute yourself.
Ma'am, it's unmute only, ma'am.
No, mute. So
Ma'am, can you
background,
hear me?
I can hear you, Ashita. You just mute
Okay, sure.
sound. Yes, it's disturbing. The background sound is there. So I can see many of new names in the group. Can you just introduce? I think Ashita, you're new, first class today. Yours is first class, Ashita.
No, ma'am, this is my second class.
Maybe I was confused with one more Asha. Asha is there.
Yes, ma'am.
कौन सा आशा हो आप? You're from our college, Asha?
Yes, ma'am.
No, okay. I identified you. Okay, bye. Bye in the sense mute yourself. So, okay, I was discussing about the types of excipient interaction also, chemical interaction and the physical interaction. And the interaction can be positive effect or the negative effect. Means interaction can be an advantage or a drawback. Okay, many times it's a drawback only more than the advantage. But there are excipients which can be deliberately added for the advantage purpose. So also we started with the physical interaction in detail. Means during the processing, when we carry out the interaction procedure in the laboratory, you may not find any interaction. But when the same thing, when we subject it to the processing, so it becomes mandatory for us to even analyze the sample in between the processing to make sure that there is no drug excipient interaction is happening. Okay, So the processing factors are also there, which results in the physical interaction. With respect to that, I discussed with the two processing factors. So I was telling also that, see, we cannot study in detail that each and every drug excipient interaction. So every drug will interact differently with different excipients. And with the same excipient, some other drug will have interactions in different interactions. So it's just an example. In exams also, when you write the points, no, you're just writing the example. You just write the examples and then the processing also can affect the drug excipient interaction. So metoclopramide hydrochloride, when it is mixed with the amorphous lactose, even the nature of the excipient is mattering here. So they have observed that amorphous lactose is actually increasing the interaction level compared to the crystalline one. Means the lactose can be of the different types, which are crystalline, hydrated form, unhydrated form, okay, monohydrated form, or in the amorphous form. So by selecting the excipient also, we can specifically select which form you want. So they have observed that amorphous form has more interaction capacity. Means that results in the Maillard reaction, nothing but a pigmentation reaction which happens. So the color change will happen. Even that is an interaction, right? Physical change also is very important for us. Not only chemical interaction is bothering us, even the physical change happening in the dosage form also should not be ignored. So amorphous form, they have observed that it's causing more of interaction, especially during the milling process, because the surface area is increasing. Because of the increase in surface area, the reaction kinetics also will increase because of the increased surface area. So what they did was they could avoid that by a process called as annealing procedure. Okay, they carried out annealing process so that they could minimize the effect of milling on the interaction. So milling has a positive effect, sorry, the negative effect because it is resulting in the Maillard reaction, means unwanted reaction. And they could do, means if for everything what we perform in the pre-formulation studies or the post-formulation studies, it's all the experimental based results only. We cannot just theoretically say this might happen unless until you have some already means published data telling that okay, lactose is causing the interaction with this drug. So maybe next time when we do with our drug of the same, almost like the same chemical structure of the already published one, I can expect the interaction. Without doing also, I can expect the interaction, that there is a chance of interaction, and I can avoid using that excipient. But initially, the trials has to be, means we need to carry out the interaction or just without doing the processing or of the processing, both. So this is how the data will be represented for us to understand what is the effect of the interactions. So yesterday, I don't know how much you understood about this. See, look at the y-axis, it is a metoclopramide. Actually, its amount is decreasing in the sense what? When I measure the amount of metoclopramide from the day 1 to the some other day after milling process, or that is after 5 minutes or after 15 minutes or whatever. So when I compare the metoclopramide percentage at the 0 time interval, as you can see at the 0 time interval, there is 100% metoclopramide because there is no interaction. But as the milling increased in this direction, as the time increase of milling, they started taking the sample in between. They could observe that there is a decrease in the metoclopramide amount, means whatever the difference is there, that is getting interacted, means it's no more the original form of the metoclopramide. That is what the graph indicates. So like this, they will do the studies in the laboratory to confirm that there is interaction happening or not. Anyway, I'll be discussing later on what are the analytical methods. See, I told you, visibly you can't make out the interaction unless until some physical change happens, like color change or the texture change. All that can be easily seen, but many of the reactions are not visible to us. So we have to somehow, we need to depend on analytical techniques. So that's another subtopic which we'll be learning in detail. Analytical techniques used for assessing the drug excipient interaction. Now I'm just briefing only the types of drug excipient interactions. So just remember some of the examples and try to understand. So interaction happens, and interaction can happen at any time. It can happen during processing also. Interaction can happen even during storage time also. Because during storage time, there is a possibility that environmental factors will affect. Change in the humidity condition, change in the packaging material, whatever we are using for the packing. So anything may be possible which can aggravate the interaction. Okay, so just with example, please relax and understand. So don't worry so much. 1 or 2 examples you remember and try to put your own sentence. So that was about the metoclopramide, which they could see that effect of milling, especially when they use lactose, that is amorphous form of the lactose. Another study also they did where to check the effect of compression force. Even not only milling, during the compression also they could observe that the amorphous form of lactose increased the reactivity. Means even the not only milling, compression also has the effect. Means these are the examples of processing effect on the interactions. So reactivity increases with the amorphous. So what they did was option was they selected the other form of lactose. Instead of amorphous lactose, they went with the spray dried lactose, which is anhydride form. Okay, other one is a spray dried lactate monohydrate form, and they could see that comparatively, if you look at this bar graph, see graphical representation has a lot of advantage. It makes us to understand with clarity. So that's the advantage of graphical representation. Initially, when they use amorphous lactose, don't look at the black bar graph, just look at the plain one. So amorphous lactose, when they use, they could see the metoclopramide as drastically reduced because of the interaction during the compression. Okay, and this when they change that amorphous lactose into spray dried lactose, what they could see is though there is an interaction happening, but it is very less. Means the change, the excipient type also is going to matter the interaction. Same thing they could observe with the spray dried lactose monohydrate. Much it's not there is not much at the excipient interaction. That is the metoclopramide and the lactose. Lactose is excipient here, and the metoclopramide is the drug in our case. So alternately, what they did was to minimize even that unwanted reaction, though whatever was very minimum, they did the annealing process, and they could observe that annealing process added up advantage. So even with amorphous lacta, after annealing process, they could see that there is a less loss of metoclopramide. As you can see, look at this levels here. This indicates the level of the metoclopramide. Okay, so annealing has an advantage. So this is how the planning goes on. See, drug excipient interaction gives us lot many ideas. It gives us the option to look for any other alternative excipients. Of course, I don't want this. Let me go with other one because this has not many drawbacks. Or I can also look into, I want to retain the excipient. I don't want to leave that. But somehow I want to overcome the reaction happening. So I can look into the alternative, keeping the same excipient. What is that I can do so that the excipient is retained, but still the reaction is not happening. So that's what they could do with the annealing process. So they can keep the same excipient, but still they could minimize the reaction happening between the drug and the excipient. So like this, you can get to see lot many examples in the textbook if you have the option to refer the textbooks. So now proceed with the next one, some of the examples. So next type of interaction is liquid on solid adsorption because of the presence of the moisture on the surface of the. These are all the physical interaction I am just highlighting about. The one physical interaction was with respect to the processing, that's the milling and the compression. The next physical interaction we are going to understand with example is the liquid on solid adsorption interaction. It is called as. In a simple way, it is a moisture. It is a moisture which is getting adsorbed or absorbed, all that. You should be knowing by this time what is adsorption and what is absorption. They're two opposite, not opposite, they're the two different phenomena which happens on the because of the presence of moisture on the solid surface. So interaction between solid and moisture is maybe due to the adsorption of the moisture as a monolayer or multi-layer, or maybe present as a condensed water at the surface. So no matter what is the thing, it's the moisture ultimately. The moisture may get deposited as a uniform layer on the drug particle, okay, or it may be getting deposited as the multi-layer. So whatever the reason it might be, so that is the moisture is the one which is actually causing the interaction between the drug and excipient. So adsorption of moisture on the crystalline solid can result in so many drawbacks. These are the drawbacks with respect to the adsorption of the moisture. It can result in the crystal hydrate. The anhydrate form can become into hydrate forms, can become deliquescent. You know, deliquescent is very difficult to handle because of the more moisture content. Capillary condensation, where the moisture keeps with the capillary action, it keeps condensing down into the particles. So water can be absorbed through the hydrogen bonding. This is just a mechanism for your knowledge. The water can absorb through the hydrogen bonding or the coordinate covalent bonds. Okay, it can form either with other water molecule or with the drug molecules. So this is how the water can play its role. So liquid on solid adsorption is another type of physical interaction. which you can expect in the drug excipient interaction. So the presence of this, what happens, the moisture can disturb many of the properties. The moisture can, we know that bulk property, especially the flow properties of the powder will be very much affected because of the presence of the moisture. So this amount of moisture absorbed depends on again of the mainly like this. If you look at the topic, how it's flowing, no, there's so many things which come, which are coming in our main topic, what we are discussing. It's we need to have a basic knowledge, strong basic knowledge, so that it becomes easy for us to correlate. So if possible, time permits, whatever the pharmaceutical subjects you had in your UG level, keep just revising once, especially the physical pharmaceutics one. And next comes, since you are into branch of Sutics, maybe that you can revise the industrial pharmacy also. All the pre-formulation studies, whatever you would have learned, or the dosage form studies you would have learned, just view it once, review it once again. So all these are the properties which are going to affect it because of the absorption of the moisture. And now this moisture is a culprit that it doesn't keep quiet. Okay, it disturbs, it causes all this unwanted effect. That's what we came to know. All this unwanted effect is caused because of this. And at the same time, now not only that, it also can cause the phase transformation. It can completely change the physical property of the drug. In the sense, phase transformation is like transferring from monohydrate to the dihydrate or anhydrate to the monohydrate. So which results in the further physical and chemical instability. Okay, and this can be aggravated in the presence of the excipient. So whatever is happening in the presence of the moisture, that can be aggravated in the presence of the added excipient also. Even that has to be kept in mind, whether the added excipient is aggravating these reactions. So what has to be done? Because indirectly or directly, it can affect the physical property or the chemical instability, even the manufacturability, and also it can affect the bioavailability of the drug. So all the details are very much important to be noted down. So what is phase transformation? The next one is the phase transformation. See, drug excipient interaction. See, drug as such may be stable or may not be stable. See, when I just look into API, active pharmaceutical ingredient, it may undergo oxidation, it may undergo hydrolysis. There are so many things which can result in the stability issues of the drug as such. We know that. It may be stable or it may, or photosensitive, for example. But how to overcome this? See, or as such API has the stability issues, added excipient can aggravate the stability issues. That is the problem. So we should be careful in selecting the excipient because already the drug has the problem. This added excipient should not add up the problem. So while selecting the excipient, we are making sure that the added excipient is not actually accelerating the stability issues of the drug. So phase transformation, just now I told, absorbed water can change the physical and chemical properties of the drug. Okay, and that mainly happens because of the formation of the hydrogen bonding or the coordinate covalent bonds. It's with respect to chemical aspect. Okay, so this complex process that involves partial dissolution of the drug under high RH condition. So it is one of the process which can disturb the dissolution process. So maybe this example will make you understand better if you just look at go through this example. See, they have observed during the manufacturing of nitrofurantoin anhydride during some formulation process, what they noticed what this nitrofurantoin anhydride is like to, for example, phase transformation of this nitrofurantoin anhydride to hydrate has occurred in the presence of lactose monohydrate or microcrystalline cellulose. In this, nitrofurantoin is the drug and the lactomonohydrate or MCC is the excipient. So what they have noticed was the phase transformation, which is an anhydrate, no water content. So whatever nitrofurantoin was added in the beginning during the formulation process, that is a manufacturing process, the lactose monohydrate was added as excipient, and MCC as the microcrystalline cellulose as super disintegrate or the diluent. Okay, they have their own rules. So this was converted, anhydrate was converted into hydrate. That was the problem they could notice, and that was more in the presence of lactose monohydrate and the MCC, and that too in the presence of high humidity condition. See now, there are so many things are adding up. It's not only the type of excipient, even the environment around the processing, even that is started interfering. Means drug excipient interaction is not a simple thing that which you can easily understand. So thorough scrutiny has to be done at every level so that any of the drug excipient is noticed, so that can be somehow you need to overcome. So same that's what I was telling you, MCC and the lactose monohydrate, they observed that it accelerated, means conversion into hydrated form was accelerated in the presence of the monohydrate, that is microcrystalline cellulose or the lactose monohydrate. Now one more example what they noticed was carbamazepine in the presence of PVP-K12. Again the PVP-K12 is the another excipient, and PVP-K90. They enhance the dehydration. So they took it as an advantage. The added excipient causes dehydration. Dehydration means what? Removing of the water. Okay, in the case of lactose monohydrate MCC, it was causing hydration. It is in the presence of humidity, it was causing hydration. So that was a drawback. But in this other example, carbamazepine, what they observed was the added PVP-K12 and the PVP-K90, though it is causing interaction resulting in the dehydration, that dehydration was advantage. So this excipient functioned as a molecular desiccant. So they decided to add this as a molecular desiccant so that the absorption of the moisture can be avoided. So that's what I was telling. The added excipient can be a positive advantage, or it can be a negative disadvantage. So in the second example, it was an advantage and they decided to add it as a desiccant so that it will not cause any kind of phase transformation. Hope at least you people are making some points, though you're not able to write entire thing, at least make note on the points whatever we are discussing. So look at the next example. The moisture, we know that it is resulting in the phase transformation, and same thing was reported in the theophylline anhydride, and this was happening more during the compaction process. So look at now the process also coming into picture. So previously carbamazepine, whatever example we just understood, okay, those are the two different examples. There also the moisture was playing its role. Same here also, theophylline anhydride was reported. Okay, we could see the phase transformation because of the moisture absorbance, and that was increased due to the compaction in the presence of the hydroxypropyl methyl, which is an excipient. So it's another processing factor we understood, which is also resulting in the excipient interaction. And not only that, even the presence of hydroxypropyl methyl cellulose (HPMC), it caused the theophylline to convert into, like, theophylline anhydride was converted into hydrate, which actually results in the phase transformation. Anhydride to hydrate is the phase transformation. Okay, that phase transformation was enhanced during the compaction. Okay, and hydroxypropyl methylcellulose (HPMC), no doubt it is causing phase transformation that was aggravated during the compaction. So compaction also has an effect during the drug excipient interaction. So these examples you just note down. So anything you would like to ask from your side? Because this chapter, where it is giving examples and making you understand the importance of drug excipient interactions. So it's like full of examples only. So the last statement, whatever you are seeing here, this part I'll be discussing little later, as I was telling you how to identify that interaction is happening. So we need to depend on the analytical techniques. All this technique in brief we'll be learning in another class in detail. So let's go to the next one, chemical interaction. All that were the physical interaction examples I just covered. Though physical interaction can result into a chemical interaction, also we saw that Maillard reaction, milling, compaction, compression process actually aggravated the Maillard reaction. So it is a physical followed by the chemical interaction. So it doesn't matter, but just as an idea I gave you, these are the physical interaction which was happening. And now these are the examples, quick examples we are just going to go through. These are the chemical interactions. And the reactions can be categorized into varieties. It can be like acid-base interactions, charge interactions, hydrolysis, Maillard reaction, transacylation, so like this. So let's go into the first one, acid-base interaction. So remember, we are not learning in detail what is acid-base interaction, how it happens, all that. Just for your knowledge, an interaction chemically can happen between the drug and the excipient, and that interaction can be of varieties. So it is superficially we are going to learn, not in depth how it is happening, all that. No. So many drugs, it's just justification point. So many drugs are either weak acid or a weak basis, and 75% are weak bases and 20% are the weak acids. These are all statistical data. So now this 45 to 50% of the marketed drugs are salt form. We know the advantage of converting the drug into the salt form because it has solubility enhancement. I mean, converting into salt will have the enhanced solubility. And majority of these are weakly acidic or weakly basic. So overall, the drug, whatever we have, most of the drug are either weak acids or a weak basis, and most of them are, many of them are in the salt forms. So when we say that, means that they are either acidic or basic in nature. So solid state acid-base reactions are common interactions. Okay, so that's what I just want to tell about acid-base interaction. So whatever points you are seeing here, this is something to do with analytical technique which is not required. Means what they are trying to say is, when we are planning to identify the acid-base reaction, we depend on the techniques, analytical techniques. And this technique should not have the solvent extraction. There should not be any procedure which actually results in the solvent extract. It's something to do with analytical technique, because this process of analysis can also result in the acid-base interaction. So look at this now. Processing, that is milling, compaction, compression, that was the processing with respect to the dosage form. This processing, what we are talking, analytical technique, even that analytical technique method, what we follow, even there is a chance that during that sample preparation for this analytical technique, though there is no acid-base reaction happening, but due to the processing, whatever we are doing for analysis purpose, can result in the acid-base interaction and give us the false positive result. So look at the means the possibility of interaction happening at what stages. It's not only during that, as I said, in the formulation, that just initial pre-formulation stage. It can happen in the during the formulation stages where the processing factor, and now in the analytical stage also there is a chance that there is an interaction happen. So just in overall, they are just trying to tell that while selecting a screening technique to identify the acid-base interaction, make sure that it will not add up from its side any kind of interaction and gives the false positive results. So that's about acid-base interaction. So I was telling you, I'll just discuss all this in the techniques in the later on. Means every interaction can be identified by the various technique, and these are some of the names of the techniques. The next one is the charge interaction. So charge is nothing but either the positive ionized drug generally will have the positive charge or the negative charge depending on the functional groups they have. So the charge can be, the charge is present even in the excipient, charge also present in the drug. So the interaction occurs in the excipients and the drug with ionizable group, mainly the groups like carboxylic acid and the amino acid, COH and the NH3, that is amino acid group, that can generate corresponding anion and the cation. So ultimately charge interaction can happen when there is a charge between the excipient and the drug. Okay, acid-base interaction based on the acidity and the basicity of the drug and the excipient interaction happen. Same here, charge interaction based on the charge. So this interaction is also interdependent not only on the type of the charge, but also it is interdependent on the pH of the environment. Because when I say drug is an electrolyte, drugs are electrolyte. Electrolytes are what? Which has capacity to ionize. Most of the drugs are electrolyte in nature. The another category is non-electrolyte. The compounds which are non-electrolytes doesn't have any change. They don't have the capacity to ionize. So, okay. So now when I say drugs, most of the drugs are electrolyte, they can ionize, no doubt, and that capacity of ionizing, all will not ionize, entire amount will not ionize, some part will remain unionized and some part will remain ionized. When I say some part is remaining ionized and some part remaining unionized, that depends on the pH. At what pH your drug is present. Depending on that pH, this ionization can occur. That's the reason the charge interaction is dependent on the pH of the environment. So what pH environment the drug is sensing, the drug in the excipient is sensing, depending on that the interaction can happen. So if the charged excipients are mixed with ionizable opposite charge drug, we know that just now that's what we are studying, it results in the insoluble complex. So it's just a mechanism what is happening. The drug interacts with the excipient because of the presence of the charge, and that results in the insoluble complex. And the insoluble complex many times can be an advantage. Sorry, when I say complex, just complex can be an advantage because the complex can be insoluble complex or the complex can be soluble complex. So when I say insoluble complex, this is the example which is a drawback. Diclofenac sodium, when they prepare the matrix tablet, and we know that matrix tablets are what? One of the modified release dosage form. So as such, it is a modified release dosage form where the drug will be released very slowly to the GI environment. So what happens? This diclofenac sodium, when it was prepared in the presence of the polymer cytosine was the polymer at the low pH. What happened? There was a complex formed between the cytosine and the, that is diclofenac sodium, and that forms ionic complex which was formed between the diclofenac sodium and the ionized cationic polymer retarded the release. I mean the release of the diclofenac sodium was disturbed or inhibited. Why? Because it is forming the complex between the diclofenac sodium was forming complex with the polymer cytosine, and that complex was formed. Why? Because they belong, means they have the charges. They have the opposite charge which results in the attraction and results in the formation of the complex. So this is the drawback because it is forming a insoluble complex. Hope you understood the complex negative effect. If you another two or one slide, maybe I'll be telling the positive effect of the same complex where it's an advantage. So this was the charge interaction. So one more example added to the charge interaction. I'm expecting you to note down these examples because the textbook what I referred, I don't, it's not there, it's not available online. So it is in the college. So I don't know when you guys will be coming to college. So better when I'm telling, note down the points. So interaction, that was first example was the diclofenac sodium with the cytosine. The next example what we will be learning is the interaction between the metformin and cross caramellar sodium. CCS, okay, which is an excipient. Metformin is a drug and cross-caramellar sodium is the excipient. And this metformin, which is a strong base, all these details not required for you. But if at all you're very good in the knowledge of pKa, you know every drug has its own unique pKa value, and that pKa value is the deciding factor depending on what pH of the environment, how much to be ionized and how much to be unionized. Means they're all the interdependent factors. pKa of the drug is the property of the drug. pH is the property of the environment in which the drug is present. Now, how much drug is getting ionized and how much drug is remaining unionized, that depends on the what is the pKa and what is the pH around it, that particular drug. So what they noticed was this pKa, the metformin was interacting with the CCS. Just an example as a complex formation. That is because of the presence of this charge group. That's what I'm trying to stress upon. But what they modified was, to avoid that interaction happening, they added one more component, arginine, a competitor was added to eliminate the interaction between the metformin and the cross-caramellar sodium. So this is a decision factor. Means because I knew there is an interaction happening between these two, that helped me to decide that I need to add a third substance so that I can avoid the interaction happening. So don't you feel that it's one of the important decision that we are making during our formulation aspect? Of course, it is an important thing, decisions which we are making. So this is how Miss drug excipient interaction not only helps in ruling out the excipient. I don't want this excipient, let me not use it. Let me take the other one. It's not only helping us to selecting the excipient, but also it is helping us to come out with the alternative. That how it can be minimized. By using same excipient, how it can be minimized. And one more example to that, also the diclofenac and the udrogit, whatever the diclofenac and cetirizine I told, the same thing they could observe it even with the udrogit polymer because of the complex formation. One or two examples better you remember. If you're not able to remember more than two, it's okay. So coming to the next type of chemical interaction, that is a hydrolysis reaction. You know what is hydrolysis? It happens in the presence of water. All that chemical theory aspect we are not discussing in detail. So the example, just look at, let's look into the example. Phosphinopril sodium, okay, which belongs to AC inhibitor, that is the category. So what they expected was when they formulated this in vivo, this it is converted into phosphinopril. That is a conversion which is must for this particular drug for to exhibit its action. And that happens mainly because of the hydrolysis. Hope you are able to understand. Phosphinopril sodium, to exhibit its action, it need to get hydrolyzed in vivo, that is inside after taking to phosphonoprilate. Okay, and that is by hydrolysis of the phosphonic acid ester side chain. That is a mechanism. Even I'm not much bothered. I'm just giving you idea why this is important for us to know. Now what they observed is during processing of this phosphinopril, that is during the formulation process, they have added the magnesium stearate for its as an excipient. Okay, that is lubricant. And this, what happened is magnesium stearate, and in the presence of moisture, what happened? This magnesium stearate is aggravated or it interacted such that it is converting the phosphinopril to phosphonoprylate, not in vivo during the processing itself, which was a drawback because this phosphonoprilate was supposed to be forming inside, not outside during the formulation. It will lose its activity. So that's the reason, means the magnesium, this is one of the interaction which showed that the addition of magnesium stearate can cause the hydrolysis, aggravate the hydrolysis reaction. Therefore, magnesium stearate should be avoided in the formulation. That's what they concluded. And this magnesium stearate, its reactivity is high in the presence of 0.5, that is microenvironment. What is microenvironment? You have any idea about what is microenvironment? I've been telling many times, I told actually this microenvironment. Soundarya SK? Trupti?
Ma'am,
What is microenvironment?
uh, only in the room or in the area which we perform.
No, microenvironment of the drug I'm talking.
Part
This
of
is
organism.
the, who is that? Tejaswini, you're telling part of organism?
Yes, ma'am.
No, dear, it is nothing to do with your microorganism. It's something to do with the area around the drug particle. Imagine one drug particle and just imagine the area, surrounding area. For example, I put a drug particle into the water. So vicinity around the drug particle is water, and water has its own pH, correct?
Yes,
That
ma'am.
is called as microenvironment. The vicinity around the like particle we are talking is called as microenvironment. So this magnesium stearate, when we do the granulation process, we know that we have many ingredients for the granulation process. But if it happens so that the pH of the environment surrounding the magnesium stearate is 5.5, it is causing the interaction. When the interaction is high in this microenvironment. No doubt magnesium stearate is a drawback, but that drawback is aggravated even in the presence of the pH of the environment. That is microenvironment, and they have observed that it is almost at pH 5.5, it is very high. So all this information definitely not going to be is overlooked. Everything will see. How come we are reading all these points? I have not done the study. Though I'm teaching you, I didn't do these studies. I didn't do personally all this study. I'm just reporting you the data, whatever I could get it from the textbooks or even from the online articles. That means they have published it. So that is making us to actually get the knowledge. Like what are the possibilities of interaction happening? So referring the textbooks or referring the online articles, it is so important. The literature, the what we call it as the literature. Please remember, all of you, it might not sound very important for you right now, but when you go to the second year, the literature information is so important when you plan for your research project. So that is one of the very important steps we need to carry out. One of the, it's very much required. So I can minimize my, see, for example, I didn't had idea about magnesium stearate drawback, and I am doing the experiment. Let us imagine, or maybe in industry they are doing the production. So let us talk in either level. In the lab scale, me, I'll be doing in the lab scale because I'm in the college administrative area. But if the same person, someone in the industry is doing with the same magnesium stearate without having this knowledge, maybe you'll end up with the loss. So, but the literature is telling you, okay, this is what happened with our work. So it's like an alarm to the person who is using the particular excipient. So literature is helping us in many ways. So this information, whatever I'm just telling you, is the literature information published in the textbook or published in the articles. So the magnesium stearate, you get to see not only is a culprit, though it is playing very important role in our tablet manufacturing process, and its percentage 0.5%. It's not more. We added more than 0.5% because less than that, maybe it is safe for us to use, but more than that, it causes unwanted effects. So magnesium stearate was excluded in this study, especially, and they went with the alternative excipient. One more example is like chemical reaction is transacylation. The example what you could see that was absorbed in the presence of starch. So they could observe that the presence of starch, and again the same magnesium stearate is coming when they were when they were trying with the aspirin formulations. Okay, aspirin, the tablet formulation containing aspirin and phenylephrine. Okay, there are two ingredients here. They reacted to give acyl compounds. It's one of the chemical compound, acylation process it is called as. And it gives acyl compounds and salicylic acid in the presence of starch and magnesium stearate, which was not acceptable. Means the acylation is one of the chemical reaction, and that acylation they could observe when they were planning to formulate the aspirin containing the phenylephrine in the presence of starch and magnesium stearate. What they noticed was the reactivity was high, especially with the magnesium stearate. Though the starch was causing the interaction, it was only 1%. But the magnesium stearate almost it resulted in the complete degradation. So that was about the acylation process is also one of the chemical interaction, and this is one of the example which you can add up to the answer point. Not just as an additional information, I'm just telling you, when you are handling the drugs which are proteinaceous in nature, which are protein. See, we know that drug products are of varieties. Okay, so proteins, peptides are also they are the drug molecules. So these proteins, as such, they are very susceptible to degradation because they're very sensitive to the pH, sensitive to the temperature. And when we take temperature, processing will add up this temperature factor because of the friction happening in the equipments, mixing equipment or the blending equipment, wherever the equipment comes, there is a chance that there is a friction happening. So during processing and in the presence of moisture, they have noticed that the protein as such is not stable, but its stability is also altered because of the presence of the excipient. That is one thing. But same excipient was also added, some of the excipients were also added, and they could stabilize the protein molecules. Okay, means this is a positive aspect I'm talking. The excipient, there are excipients which can cause the stability issues of the protein, but there are excipients also which could be added deliberately in order to cause the stabilization of the same drug. And sucrose was the one of the example. It increased the stability of the protein trypsinogen. It's not a common excipient that you can add for all the protein drug molecules. First, one of the example is trypsinogen protein, which stability was enhanced by the presence of sucrose, which is an excipient. And this protection of the protein trypsinogen, which was happening in the presence of sucrose, that is mainly because of interaction only, but it's a positive interactions. So like this, these are some of the examples of the chemical interaction I just highlighted now. Translation, hydrolysis reaction, charge interaction, acid-base interaction. So these are some of the and the phase transformation. The one thing left out is the Maillard reaction. Though I was kept on telling you what is Maillard reaction, it's nothing but the reaction happening between the drug and the carbohydrate. So carbohydrate is the main component here. Any excipient which are nature of like carbohydrate nature, and also some of the amines, they produce the brown pigment because of the interaction. So, okay, and this carbohydrate may be like glucose, maltose, and lactose. That's the reason the Maillard reaction in case of metoclopramide during milling and the compression, it was enhanced, aggravated in the presence of the lactose monohydrate. So got the reason now, because it is a Maillard reaction, is a reaction which is named for happening between the carbohydrate and some of the amines with respect to the counterpart, that is the drug molecule. So these are some of the examples: fluoxetine hydrochloride with lactose. Already metoclopramide you have noted down, even that can be the one of the example here. So where they use the spray dried lactose. And the reaction rate also depends on the mixing mode. We know that even that milling process can also result, enhance, aggravate the Maillard reaction. And also this was increased in the presence of magnesium stearate. So throughout your drug excipient interaction, the one repeated excipient name you'll be hearing is magnesium stearate. Okay, so it is like, though it has advantage of adding magnesium stearate, but as I said, percentage is very important. So this was about the chemical interactions examples. Anything you want to ask me? The last one, solid on solid adsorption. I think this slide was supposed to be there. I don't know how it has come here. Solid on solid adsorption process. It means interaction can happen. The physical interaction, this is an example of the physical interaction supposed to be, actually not as a chemical interaction. So this magnesium stearate I was telling is the major culprit again. This actually, when we add it as a lubricant, percentage is important, I said. If you add excess, what happens is it forms the coating around the drug molecule. Okay, so physically it is actually causing the interaction, means the drug will get coated unnecessarily by the magnesium stearate and it will disturb the dissolution. It will not allow the drug to undergo dissolution. And also it can result in the excess of magnesium stearate when you result in the unmixing. Mixing and unmixing, they are counterpart. Unmixing results in the segregation, and the segregation results in the disturbance in the drug uniformity. So it's again interdependent sequence. So this magnesium stearate as such can retard the dissolution. Okay, so what they understood in one more process was when they added SLS. This SLS causes interaction with the magnesium stearate and it pushes out the magnesium stearate from its place. So it's an interaction, but there is a positive effect. This prevented the deleterious effect on the dissolution. In the sense, we know the magnesium stearate has this drawback, the statement, the blue statement, whatever you're reading. But when they added SLS along with that during for some formulation, what they noticed was even the SLS was interacting, which results in the freeing the magnesium stearate. Okay, though it is an interaction, because the magnesium stearate was freed, that prevented the unwanted effect of the magnesium stearate on the dissolution. So it is solid on solid adsorption. Like liquid on solid adsorption we learned, where the moisture deposits as a monolayer or a multi-layer on the solid surface. Same, this one is solid on solid adsorption, where SLS is getting deposited on the solid particle, that resulted in the release of the magnesium, freeing of the magnesium stearate. So that's one example of the solid on solid adsorption. So anything you want to ask from your side? Last, just before I wind up today's class, just some examples I want to stress upon. Where deliberate interactions, we try to make the deliberate interaction between the drug and excipient. You can mute yourself for another 1 minute. So interaction can be positive or interaction can be negative. That's what I kept on telling. So these examples, what two example I just quoted here. Then maybe the second example is a common example which you know, where they use the cyclodextrin complex, formation of this complex drug cyclodextrin complex. Danazole may be one of the drug example. If you can't remember the drug, it's fine. You can say complex formation between the drug and cyclodextrin, and the cyclodextrin is an excipient. The complex formation between this, we understood in the long back, some of the slides, where the diclofenac sodium interacted with the, which is that, I forgot that excipient. It results in the poor dissolution. It did not let the dissolution of the diclofenac sodium because of the complex formation. But here, the cyclodextrin interacted with the drug results in the enhanced solubility. So it is a deliberate interaction example. So because cyclodextrin is an hydrophilic carrier, balanced hydrophilic lipid carrier, which is more of hydrophilic functional group is that it is cyclic in nature, non-reducing oligosaccharide it is. Okay, there is no chance of Maillard reaction to happen, and it is generally used as a carrier. So it forms an inclusion complex that results in the enhanced solubility. So this is a deliberate interaction example. Same with this bucomerol magnesium oxide interaction. This is used intentionally for positive changes, okay, such as increased plasma level due to faster solubility. Okay, there's another example where the deliberate interaction was done. Interaction between bucomerol magnesium oxide interaction resulted in the increased solubility, and this is mainly because magnesium chelation with the bucomerol. The chelation process happens, which causes increase in the pH of the microenvironment of the drug, and because there's a change in the pH, the solubility is dependent on the pH. So the pH was favorable for the solubility, and that's how the bucomerol solubility could be enhanced. So these are the examples of the deliberate interaction process. Okay, then if you want to ask any doubt, you can ask me before I wind up the session. Are you all noting down the points? Trupti, you are first class today.
No, ma'am, second class.
ನಿನ್ನೆ ಇದ್ರ ನೀ ಮೊನ್ನೆ
ಹಾ ಇದ್ದೆ ಮ್ಯಾಮ್.
ನನ್ನ ಕ್ಲಾಸ್ ಅಲ್ಲಿ.
ಹಾ ಮ್ಯಾಮ್.
Okay, it is T for Trupti. I thought T for Tejasvi. Tejaswini is TS, it is showing.
Yeah.
Okay. Anyway, this go-to meeting we are taking classes. The advantage is your attendance will get recorded automatically. So what, even it gives us what time you entered, what time you exited out of the room. All the small details it gives. That's the advantage of go-to meeting, and that's the reason our college, our principal sir is insisting to
Excuse
use
me,
the
ma'am.
go-to. Yes, tell me, dear.
मैम बायोफार्मास्यूटिकल इंटरेक्शन ये जो पॉइंट है ये ड्रग एक्सपिएंट इंटरेक्शन में इन्वॉल्व नहीं हो सकता है क्या?
मतलब I didn't get your question.
बायोफार्मास्यूटिकल इंटरेक्शन ये जो टाइप ऑफ ड्रग एक्सपिएंट इंटरेक्शन.
Are you referring to the syllabus by chance?
Type of drug excipient interaction.
They ask you type of drug excipient interaction, nothing but that positive interaction, negative interaction, physical interaction, chemical interaction.
And ma'am, biopharmaceutical interaction?
It's nothing but same.
Okay, and excipient interaction is same, ma'am.
Excipient, see, interaction can happen with anything. That's what I was telling you. It can happen between two drugs, it can happen between two excipients, it can happen between
Yes.
drug and excipients.
Hmm.
Okay, so interaction can happen anything for any reasons.
Okay,
So since
okay.
as a syllabus, it is mainly the drug excipient interaction we are highlighting. So we are trying to understand why we need to study that, or why we need to know that there is an interaction happening. Even the excipient interaction is also not acceptable. Even that is dangerous. Correct.
Okay.
Anything can go wrong with the excipient interaction also. Means overall, we are learning about interactions, okay, and why we
Hmm.
need to know that interactions, because we can avoid unwanted expenses or the wastage of the time happening.
Okay.
So that, and ultimately it's a stable product. We should get a product, tablet or a capsule or a syrup or an emulsion. It should be stable ultimately, and it should give the required therapeutic action. So this interaction is such a thing that it may not happen only during processing. Even it can happen, that's what I was saying, even it can happen during storage also. When we don't follow the instruction of storage conditions, interaction can happen at storage conditions also. Maybe that it would have qualified in the during the QC test, all that. But during at home, when if you can't preserve it properly, there's a chance that interaction can happen, which you can't make out. Okay, biopharmaceutical interactions are nothing but all these interactions.
Okay, okay, ma'am.
Okay, interaction is a word, something is like another big ocean, dear. We can talk about drug interaction, another aspect where the drug will interact with another drug, it will disturb the therapeutic action of the another drug, like this.
Okay.
So interaction in vivo also can happen inside the body. Interaction can happen outside the body, that is in vitro. So whatever we are learning, these are all in vitro interactions, which happens during formulation, during the formulation procedure.
Okay,
Okay,
okay.
okay,
Okay,
dear.
thank you.
Okay, thank you. It will be nice. I'll be very happy if you guys interact. Otherwise, मैं गला फाड़ फाड़ के बोलती हूँ इस तरह, पता नहीं आप लोग क्या कर रहे हो इस तरह. I have to just imagine, okay, Trupti is looking beautiful today, or Saundarya is making her face. I know them, that's why I'm telling. Alva Trupti?